Welcome to the podcast, Stories from the World of Science. Our mission is to explain science in a simple and fun form that is open to all.
All right, everyone, get ready, because today we're taking a deep dive into this year's Nobel Prize in Physiology or Medicine. And let me tell you, this one's a real humdinger.
Yeah, we're not just talking about a new drug or a fancy surgical technique here. This prize goes to a discovery that's fundamentally changed how we see the very building blocks of life itself, our genes.
We're talking about a whole secret language going on inside our cells, a way for them to manage those instruction manuals, those DNA strands, in a way no one ever imagined.
And it all started with these two scientists, Victor Ambros and Gary Ruvkun, just doing their thing, studying some pretty unassuming creatures, actually.
Oh, you mean like those lab rat scientists are always messing with?
Not quite. Think even smaller. We're talking about C. elegans, tiny little worms, nematodes to be precise.
Worms, huh? Not exactly the first thing that pops to mind when you think Nobel Prize-winning science.
I know, right? But these little guys held a secret that would rock the world of genetics. And it all boils down to this.
Yeah.
Think about your body as a giant city, right?
Yeah, I could see that.
Yeah.
Bustling with activity, different parts all working together.
Exactly. And each cell in your body, every single one, from your brain cells to your blood cells, they've all got the same set of blueprints, the same DNA.
Okay, that makes sense. But then how does a heart cell know to be a heart cell? Why doesn't it just decide to become, a nose cell instead?
That's the million-dollar question, isn't it? That's where gene regulation comes in. See, before this discovery, we had a pretty good handle on how genes got turned on and off.
Oh, like a switch, right.
Nice.
Flip it on, the gene's active, flip it off, it's silent.
Yeah, kind of like that. We knew certain proteins acted almost like conductors in an orchestra, telling specific genes when to start playing their tune, making the proteins they coded for.
Okay, I'm following. So what's so groundbreaking about this whole worm thing? Did they find some new kind of on-off switch?
Oh, it's way more sophisticated than that. What Ambros and Ruvkun stumbled upon was a whole other level of control, not just on or off, but imagine dimmer switches for your genes.
Dimmer switches, so like fine-tuning the activity.
Exactly. They discovered these tiny molecules that could actually tweak how much of a protein a gene was making. And they found this, of all places, while they were trying to figure out what was causing developmental defects in these worms.
You're kidding? So these little worms held the key to a whole new understanding of how our genes work. That's got to be one for the science history books.
No kidding. Ambros was studying this one gene called Lin4, and he found something totally unexpected. Instead of containing the code for a protein like a normal, respectable gene, Lin4 was making this tiny snippet of RNA.
RNA, isn't that like the messenger molecule, the one that carries instructions from DNA to the protein-making factories in the cell?
Exactly, but this RNA was different. It didn't seem to be making anything. It was just this little piece of genetic code floating around, and nobody knew what it was doing.
Sounds kind of like a scientific mystery. Like, what's this little piece of RNA up to?
Meanwhile, Ruvkun was busy unraveling the mysteries of another developmental gene called Lin14. He knew something was controlling this gene's activity, but he couldn't figure out what.
So they both had different pieces of the puzzle, but didn't realize those pieces even belonged to the same puzzle until
they started comparing notes, and boy, when they did that, sparks flew. It turned out that the short RNA sequence Ambros found in Lin4, it was a perfect match for a part of the Lin14 gene that Ruvkun was studying. Talk about a coincidence.
Wow. So they realized that this tiny bit of RNA from Lin4 was actually controlling the activity of Lin14. But how?
That's what they had to figure out, and what they discovered was mind-blowing. These little pieces of RNA, which they later called microRNAs, they were acting like, I like to think of them as genetic sticky notes.
Sticky notes. Okay, now you've got to explain that one.
Imagine this. You've got your DNA, right? Your genetic instruction manual, and then you've got RNA, which is like a messenger, taking those instructions from DNA to the protein-making machinery in the cell.
Right, got it.
So these microRNAs, they were attaching themselves to the messenger RNA, and depending on where they stuck, they could either block the message entirely or just tweak it a little.
So they're like little regulators, fine-tuning gene activity.
Right.
That's wild.
That's exactly what they are. Tiny but mighty controllers of our genes. And the craziest part, they weren't just random sticky notes causing chaos. These things were incredibly specific, each one targeting a particular set of genes, influencing everything from how cells grow and divide to how they talk to each other. Now, at this point, you might be thinking, okay, cool, worms have these microRNA things, but what about us humans? Are we running on worm time too?
Yeah, I gotta admit, when I first heard about this whole thing, I thought the same. Tiny worms controlling their genes with even tinier molecules. It's fascinating, but are we really that similar?
That's the thing, right? It turns out this whole microRNA discovery, it wasn't just some weird worm quirk. It blew open a whole new universe of possibilities for understanding human biology too.
Oh, really? So how did they figure that out? Did they start dissecting human cells looking for tiny sticky notes?
Well, not exactly. But a few years after they found those first microRNAs in C. elegans, Ruvkun's team, they discovered another one called LET7. And get this.
What, they found it in people, too?
Bingo. But hold on, it gets even better. LET7 wasn't just in humans. They found it in a whole bunch of other animals, too. Flies, fish, you name it.
Okay, so this little piece of RNA, this LET7, it's showing up in all kinds of different creatures.
And not just showing up, I'm talking practically identical in its sequence, whether it's in a worm or a human.
So it's like what? They all got together and shared the same instruction manual?
It's more like they all inherited it from a really, really old ancestor way back when life was just getting started. And that tells you something important.
Let me guess. It means this LET7 thing must be really important, right?
Exactly. It's like finding the same ancient tool in civilizations all over the world. You know, it must have been crucial for survival if it's been passed down and preserved for millions of years. And that's just one microRNA.
So how many of these things are we talking about?
We're talking thousands of them, each potentially controlling hundreds of different genes. It's a whole hidden network, a whole other layer of complexity that we never even knew existed before these tiny worms came along.
Wow, that's mind-blowing, but also kind of overwhelming. So with all these microRNAs floating around doing their thing, what happens if something goes wrong with the system?
Well, that's where things get really interesting. And to answer that, let's talk about Dicer-1 syndrome.
Dicer-1 syndrome. Sounds kind of scary.
It's a real condition, and it perfectly illustrates just how important these tiny molecules are. See, Dicer-1 syndrome is caused by mutations in a gene, you probably guessed it, called Dicer-1.
And what does this Dicer-1 gene do?
It's responsible for making a protein that's like a molecular tailor. Its job is to snip those long strands of RNA into those perfectly sized microRNAs. Think of it like cutting fabric for a perfectly tailored suit.
Okay, I'm picturing it. So if the tailor scissors are dull, the whole outfit's going to be messed up?
Precisely. If Dicer-1 isn't working properly, the entire microRNA production line gets thrown off. And because these little molecules are involved in so many different processes, especially things like cell growth and development,
Things can go haywire pretty quickly.
That's right. People with Dicer-1 syndrome have a much higher risk of developing certain types of tumors, particularly in their kidneys, thyroid, and ovaries.
Wow, that's a serious consequence for such a tiny malfunction. It's kind of wild to think that just a tiny little hiccup in this whole microRNA process can have such huge consequences for our health.
Yeah, it really makes you appreciate just how intricate and interconnected everything is inside our cells, and how much we still don't know.
Totally. Makes you wonder what other secrets these tiny molecules are hiding. Like, if a little glitch can cause so many problems, what if we could actually harness their power for good?
Now you're talking. That's the million-dollar question that's driving a whole wave of research into microRNA-based therapies. The idea that we could actually use these molecules to, target specific genes, maybe even fix some of those faulty instructions. It's incredibly exciting stuff.
Wait, hold on. So you're saying we could, create synthetic microRNAs? Like design our own tiny sticky notes to go in and shut down the bad genes? Or maybe even give the good ones a boost?
That's the general idea. It'd be like having a microscopic repair crew that could go in and tweak the genetic instruction manual itself. Of course, it's not quite that simple in practice, but the potential is enormous.
Okay, but we're talking about, editing genes here, right? How close are we to actually being able to pull that off?
It's still early days, for sure. But the progress we've made, even in the last few years, is astounding. Researchers are already looking at microRNA-based therapies for all kinds of diseases. We're talking everything from cancer and heart disease to viral infections, even neurodegenerative disorders like Alzheimer's.
Wow, that's amazing. Instead of just treating the symptoms, this is about going after the root cause of the problem.
Exactly. It's about using the power of these tiny molecules to, fine-tune the very processes that govern our health. Imagine a future where we can treat diseases the same way we adjust the settings on a control panel, turning down the volume on inflammation, boosting the production of a vital protein. The possibilities are truly mind-blowing.
And to think, It all started with a couple of scientists and their tiny worms. Who knew those little creatures held the key to such a powerful mechanism, a mechanism that's at work in our own bodies right now?
It just goes to show the power of curiosity, And the unexpected places scientific discovery can lead. We've come a long way from thinking of our genes as a static blueprint. Now we know it's more like a dynamic conversation happening inside every single one of our cells. And we're finally starting to understand how to eavesdrop on that conversation, maybe even participate in it a little.
And what an incredible conversation it is. It really makes you wonder what other breakthroughs are out there waiting to be discovered, all thanks to these amazing minuscule molecules. So the next time you hear about some big scientific discovery, take a moment to thank those tiny worms, because they might just hold the key to the next big medical breakthrough.
And hey, for anyone listening who's feeling a little overwhelmed by all this talk of genes and molecules, just remember this. Every single one of us were talking testaments to the power of this incredible system. Trillions of cells working together in perfect harmony, all orchestrated by those tiny, mighty microRNAs. It's a symphony of life happening inside each and every one of us.
Beautifully said. And on that note, we'll wrap up our deep dive into the fascinating world of microRNA. Remember, folks, knowledge is power, and sometimes that power comes in the smallest of packages. Until next time, keep those minds curious and those questions coming.
You just listened to the podcast, Stories from the World of Science, written by Sasho Dolines.